But she announced it.
Okay, let's go ahead and get started here. So today I'm about 2/3 of the way through your exam, so I'll try to get them to you by Wednesday. Let's talk about what we have going on. We'll have a quiz on Friday. We'll have the usual office hours and help sessions this week. I'll announce them as we get closer. But today is Monday. Monday office hours are 1:15 to 3:15. If you can't make the office hours we've got, and things like that, let me know. I can always meet people on Google Meet and things like that, or we can work out other hours. My tomorrow's, we'll have some time. I'm on campus tomorrow. So, let's see, we have lab this week. And your pre-lab is to download some sort of sequence analysis software. So there was Chromis, there was, I don't know, there was something that like works on Max, there's something like 4 bases or something like that. I would just do a Google, and you want something that's going to be able to read those sequencing chromatograms. So pre-lab is download sequence analysis software. We're going to use it in lab. Okay. Questions on things we're doing? All right. So then what we're going to be talking about, we're moving into lipid metabolism. All right. And so mainly we're going to be talking about free fatty acids. Now, free fatty acids come from triacylglycerols. So your body stores your free fatty acids as triacylglycerols. So we've got a glycerol molecule, right, that would have hydroxyls on it, except I'm going to stick free fatty acids on it. Okay, and they don't have to all be the same. Maybe some will have double bonds. I think that, yeah. I think that's okay. All right. So we might have that sort of thing. And then these in your body, so these are being carried around in, you hear about LDL, HDL. Okay, you have in front of LDL, you have a very LDL. So it's a low density lipoprotein. You have very low density lipoproteins. Those become your LDL. We'll talk about that more later, but they have a lot of free fatty acids in them that are going to get extracted, and you're going to have an enzyme called lipase, and it's going to take 3 waters, and it's going to hydrolyze those free fatty acids off. So I should have made them all the same because it'll be easier to draw here. Now I have to draw 3. Let's pretend this is the top one. Okay, I may have messed up on the carbons. And then we have one that has a double bond somewhere in the middle, like that, about that. And then we have another one that had 2 double bonds. Maybe that's what it looks like. And then plus we also have our glycerol left over. I mean, the glycerol is good for energy as well, but we won't talk about anything about the breakdown of glycerol. Glycerol there are pathways for that, but we are going to focus really mostly on the breakdown of these fatty acids, and mostly the saturated fatty acids. We'll talk about what happens when it's unsaturated and how the body or how cells respond to that, but we won't really, you won't really be responsible for that. All right, so along those lines though, so we have our free fatty acids, right, and they're attached to make triacylglycerols, all right, but you can chemically do something like this. How is this different from a triacylglycerol? There's more than three, and how are there more than three? How do they manage that?
It's an acetyl group.
It's not an acetyl group.
Where's this A? Where did you find this?
You find potato chips. I'll explain it in a minute. But so for up here, what are we linking the free fatty acids to? Glycerol. Down here, what are we linking them to? Generally or very specifically?
Glucose.
There's a glucose right here. What's that?
Fructose.
Fructose, which makes sucrose. Yeah, so this is just table sugar, which food chemists have used they attach a bunch of free fatty acids to that. And this is called olein, which you may or may not have heard of. Okay, is the name of this. It's a synthetic lipid, we'll say. Synthetic lipid with a sucrose core instead of glycerol. All right, and this is something that they put into foods instead of triacylglycerols. It has the same taste and mouthfeel as triacylglycerols. Okay. However, lipase cannot release, lipase, that enzyme we just saw, cannot release the free fatty acids, so your tissues can't take it up. It can't be taken up across the intestinal wall. So it does not cross. Okay. And it does not serve as a substrate for lipase. All right, so the thought here is this is a great thing you can put into potato chips, and you will, for losing weight, right? You make low-calorie potato chips because you're putting this in, but there's no calories in them because you can't actually digest them. However, there are some side effects. Okay, one of the side effects is, well, diarrhea, which is unpleasant. These also sequester lipid-soluble vitamins.
Why is diarrhea? What's happening?
What was that?
Why is diarrhea one of the side effects?
I don't know. It's got to be drawing water into the intestine for some reason. Okay, but I don't know what that side effect is. I can explain why it sequesters lipid-soluble vitamins. This is essentially going to go straight through you, right? And the vitamins are going to like hang out with it. They're going to rather be in this. If you have a fat globule made of this, they're lipid soluble. They're going to hang out in that rather than go across your intestinal wall. And so what they do is the solutions for that are don't add too much of it to foods. And then the second thing is actually add fat soluble vitamins. Okay, and our lipid soluble vitamins, if you remember, are E, K, D, and A. Okay. So this is an interesting attempt by industry to come up with something to make things low-cal but still taste great. Okay. Not entirely successful. it's better than the weight loss solution that was on your test that I explained on your test, which is dinitrophenol, which takes things, it actually goes in your mitochondria and it breaks down that, you push protons to one side of the membrane and it just grabs them and pulls them back across. So it can go across the membrane. It can protonate and deprotonate. So it destroys that barrier. That was a weight loss attempt in the 1930s, but it causes blindness and fatal fevers. So they kind of discontinued it. But bodybuilders still use it. They try to titrate it in, burn off, make sure they have only lean muscle mass. But you have to be, I wouldn't. I used to have a fun set of videos from a bodybuilder in Los Angeles, and YouTube eventually took them down. But he was talking about using it and stuff like that. Okay, so let's talk about overall metabolism for our triacylglycerols. So what we're going to do is we'll start up here with triacylglycerols. And you could break those down. That's going to be, what would you call that general reaction that uses the lipase? Hydrolysis. Okay. If we think about dehydration, I mean, maybe, but we're going to call it hydrolysis. And then this would be condensation. Okay. And that's going to give us free fatty acids. So these are our monomers, right? We talk about polymers. When we can do lipids, they're not exactly polymers, right? Maybe oligomers, they maybe even qualify as oligomers, but so free fatty acids. Okay, we can take those free fatty acids and we can break them down, and that process is called beta oxidation. And we're going to talk about that in detail today. And then the other side is just going to be called it's fatty acid synthesis. Which is boring but easy to remember. And out of this, we're going to get acetyl-CoA. So unlike glycolysis, where we get our final product out of our pathway, and then we have to have another step with the pyruvate dehydrogenase complex to get to our acetyl-CoA. Acetyl-CoA pops straight out of beta oxidation. And then catabolism is convergent. We're going to take that acetyl-CoA and we're going to run it around the citric acid cycle. We'll have 2 carbon dioxides come out. We'll have per acetyl-CoA, we're going to get 3 NADH, 1 ubiquinol, and 1 GTP. Okay, up here in beta oxidation, we're going to be getting products off as well. We'll be getting ubiquinol and NADH out here. And how much we get depends on how big that fatty acid is. And then regardless, well, not the GTP, but regardless of where those come from, they're going to go into the electron transport chain, so we're going to end up with NAD plus or ubiquinone. They don't enter at exactly the same point. They'll go through the electron transport chain. We pump our protons across that mitochondrial membrane, and then those protons come back in, and that causes us to make, that powers our ATP synthase to make ATP. Questions on the overall? We can view out here and look at the whole thing real quick. Okay, similar to when we were talking about carbohydrates, but we have some new pathways in here, right? That's where we're really going to focus our attention. And let's see. This, I'm going to be really careful about where this, I guess I need to make that acetyl-CoA. Let's split the acetyl-CoA in half because we're going to have it in both compartments. This is going to happen in the cytosol. Okay. And this, note that fatty acid synthesis is not happening in the cytosol. This is going to happen in the mitochondria. Okay? Questions?
Why is it half of your acetyl-CoA?
I did half acetyl-CoA. You're going to need acetyl-CoA both in the mitochondria and both in the cytosol because of this process. And so the use of that is now we have a compartmentalization. So before we had glycolysis and gluconeogenesis, they happen in the same compartment. So we have to turn one off and one on using heavy signaling, right? There's signaling involved here as well, but if we have our fatty acid synthesis happening in the mitochondria and we have our beta oxidation happening in the cytosol, hold on. No, I'm sorry. Opposite. I mean, this is this and this. Oops, still wrong. This and that. Sorry about that. Beta oxidation happens in the mitochondria. But if I don't want to break down fatty acids, I just don't send them to the mitochondria. Okay. And if I don't want to make fatty acids, I don't send the reagents that I need to make fatty acids out into the cytosol. Okay, which is not, you're not going to start with acetyl-CoA exactly for that. We'll talk about that when we get to it. It uses acetyl-CoA. Questions on that? Okay. All right, so let's talk about how we get fatty acids into, well, actually, let me write out the thing about compartmentalization real quick. So beta oxidation, let me make it bigger. Beta oxidation and fatty acid synthesis occur in different compartments. All right, so regulation can be achieved by denying a pathway of the reagents it needs. So one pathway can be regulated independent of the other. By denying it the reagents. Okay, so for example, free fatty acids cannot be broken down if they're not in the mitochondria. Okay, questions? All right, so then if you're going to break down fatty acids, what you have to do is you have to prepare them. You have to get them ready. You have to stick them on acetyl-CoA and make what's called an acyl-CoA. And so the first part of that is if we look at a fatty acid, let's call that our free fatty acid. If I'm going to stick that on a CoA, I'm going to need to get that oxygen to leave, right? Okay. This is going to be a lousy leaving group. You could protonate it and be a somewhat better leaving group, but still not a great leaving group. And so what do we generally do? We stick something else on. What do we generally stick on? Something from ATP. Okay, in this case, we're going to stick an AMP on. So let's see. We're going to draw the whole structure. Okay, and they are actually ribo. When you think about just ATP, that's the energy currency. It is the ribo version. All right, so here we have ATP. And I'm going to do a nucleophilic attack. I've got some nice electrons on there, right? I mean, this is pretty stable. It's going to happen in an enzyme anyway. But let's say I do a nucleophilic attack from here onto one of these phosphates. Which phosphate should I do my nucleophilic attack onto if I want to have an AMP?
The middle one?
What was that?
The middle one?
Not the middle one. This one, right? Because I'm going to make my attack there. So what I would do is I'd have a pair of electrons, let's say, once again, stable, but it's going to happen in an enzyme, which will make this happen. I guess can come around, I'll attack there, and then these electrons are going to pop over there. Okay, and so what I'm going to get, I will not draw the whole thing again. I'm going to write AMP, right? I'm going to end up with just the monophosphate attached with the tail, and plus pyrophosphate. Here, I'll draw the whole structure for that. All right, so I've got a pyrophosphate. What happens with that pyrophosphate? Breaks into two phosphates immediately, right? The pyrophosphate breaks into two phosphates. I'll draw them like that. Maybe I'll just draw them out. There'll be a hydrogen on somewhere there because that's too much negative charge for whatever pH we're going to. We get two of these. What's the purpose of this part?
Which part?
Why break that pyrophosphate down to two phosphates? Why does that happen? Why have we seen that over and over and over again? What was that? It's spontaneous. It's going to add free energy, right? So the standard free energy change for this reaction is negative 19 kilojoules per mole. So that's 19 kilojoules per mole of driving force. These are coupled reactions, right? Of driving force to make this happen. Another way to think about it is you could think about it in Le Chatelier's principle, right? This is really low because it immediately gets broke down whenever it exists, so we drive any reaction involving it to the right. Does that make sense? Okay, so we're not done seeing that. That happens in a lot of places. How many ATP equivalents, right? How many ATP equivalents did it cost to put this thing on it? Well, actually, we haven't done it yet. Sorry. I guess I'm going to go this way. Okay, I'm going to bring in a CoA. Okay, remember the CoA has an SH on it. All right, and so the SH, we could take the H off. Well, I'm not, yeah, I'll do it. So we can take the SH off, or the H off. Imagine that, and we have these electrons come in and attack here, right? You kick the electrons up to the top, you kick them back down, you kick out your leaving group, which is AMP. AMP is a great leaving group because it's nice and stable. And what we're going to end up with is CoA attached to our, oh, it's a sulfur attachment. CoA attached to our fatty acid.
What are the arrows drawn?
What are the arrows there? That's the mechanism.
Well, yeah, but just like.
It's a crappily drawn mechanism. How about that? So we could skip, because it may not even happen in the enzyme. You may not kick the electrons up and then kick them down, though my my biochemist sense of all the reactions I've seen says it does. But so this arrow comes in here, we attack the carbonyl, right? There's multiple steps here. Like remember for your
You just have to keep it up.
yeah, so we kick them up like we did in organic. We kick them back down in the next step, and when we kick them back down, we kick these out and stick them on there. Yes.
Why, like, you trying to show with this? Like, why do we care?
Why do we care? How many ATP? So how many? This is how we're prepping this to move from the cytosol into the mitochondria. How many ATP does it cost? 2, because we have 2 phosphates. We go from ATP equivalents. We go from ATP to AMP. It's 2 ATPs worth of energy.
Turn from ATP to AMP.
I'm going to count that as 2 ATP equivalents. So we have ATP goes to AMP, 2 ATP equivalents of energy have been used. All right, so it costs 2 ATP essentially to bring to stick this stuff on CoA, okay, which is going to be important. I haven't shown you how it gets brought into the mitochondria, but it's going to be important for bringing it into the mitochondria. Okay, so this is the process of bringing it into the mitochondria. So the free fatty acid is going to be attached to a carnitine to bring into the mitochondria. All right, and that's shown in this figure over at the left. And if the question is, why do we care here, or why do we care above, is that there are enzymes involved here that there are disease states involved with lack of carnitine, lack of recycling carnitine. And so if you can't take all fatty acids in because you don't have enough carnitine, what do you think the symptoms of that would be?
What is
Carnitine is that. It's a carrier that the carnitine transporter is going to recognize as a handle and use to bring fatty acids across the membrane into the mitochondria.
Oh.
Yes.
So that last step was making it that acetyl-CoA.
The last step, everything we saw was getting to here.
Okay.
And the R is a fatty acid chain.
Yeah.
So the carnitine then gets attached.
I'm going to walk through the steps.
Oh, sorry.
That's okay. Okay, so what the first step here is that the acyl group is what the fatty acid, okay? The acyl group is going to get transferred from the CoA to a carnitine. Let's just say the free fatty acid. The free fatty acid okay? Any energy spent? See any energy spent there? Nope, I don't see it either. Okay, second step is the carnitine transfer is going to transfer the loaded carnitine into the mitochondria. Let's say moves. Okay, so we're here. We're in the mitochondria now. Now the acyl carnitine or the free fatty acid carnitine is going to transfer that free fatty acid back over to a CoA. Okay, and then finally, the carnitine transfer is going to move the carnitine back out into the cytosol. Moves the unloaded All right, so this is how we're going to get those fatty acids in. And so if you had conditions in the body where you did not want to break down fatty acids, what could you do? Where could you stop this process? You could not load it onto the carnitine. There's an enzyme involved there. You could shut down the carnitine transporter and not let things move through. That'd be two great places to do it. Okay. Any energy spent in this process? No, thankfully, right? Okay, so in terms of what I want you to know is I want you to you need to know the basic cycle above. You don't need to draw it. But you do need to recognize carnitine. Name it. Identify it as loaded or unloaded. Draw the other form.
So this protein is just like a transporter in the mitochondria that goes through the fatty acids?
Correct. And I'm guessing it's in the inner mitochondrial membrane. Recog. I think she's going to be okay. Oh, I'm still not spelling it correctly. Correct. Having a trouble with spelling today. All right. So, but yeah. So these are sites of regulation for this. And then once it's inside, then we can do beta oxidation. All right, so beta oxidation's here. I meant to print one out for you guys. Actually even made the thing. Maybe I'll do that real quick. I was actually checking to see if we have enough competent cells. I need to order some. There are a bunch of boxes, but they're all full of other stuff. Print. 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 Let's see. So you guys can take a break, take a quick break, stretch out while I go and get these from the printer. And then it'll be easier to follow along while we talk about it.
Write it down.
The fatty acids are coming down. Actually, no, less. But in the such a nice.
Yeah.
Okay. For what?
Yeah.
It's like 4 pages long. You're like,
Something like just like scratched my throat and all of a sudden. And my water bottle was missing. And so I didn't have anything to do. So I was kind of just suffering. The worst part is that like, this is all just birthday bullshit. It's just my body trying to get like the congestion out. I feel a lot better. I don't sound a lot better though. But no, I'm good now. Really? I wish it would.
It's a prolonged job.
I wish you could give them context about what he's talking about.
10:30 p.m. to 8:30 a.m.
Oh, hell!
Friday night, Saturday.
Wait, what is it? 10:30 p.m. to 8:30 a.m.?
I'll be off every other day except for Wednesday.
Okay.
I don't care if I didn't.
So this is, I'm handing out as a printout for beta oxidation. I'm going to grab one of my own and get these. So beta oxidation is a process where where a fatty acid is essentially trimmed down, 2 carbon units are clipped off of it, and in the form of acetyl-CoA. So if we go ahead and come back here, we're not going to do that part yet. Just in general, if I have a fatty acid that looks like this, imagine it's attached to a coenzyme A. I'm going to go ahead and do the simplest form here and do something that it's going to have an even number of carbons. Most fatty acids, natural fatty acids, have an even number of carbons. And then they might be saturated or unsaturated. We're going to take the simple case for now, but I've got 2 carbons here, right? So I can cut there and 2 carbons would come off. I can cut here, I can cut here, I can cut here, I can cut here, all the way down. This is a 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, 22 carbon fatty acid. Okay, and so it would take 10 rounds to cut it up into pieces. Okay, one less than it divided by 2. Let me count, make sure I did that right. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Okay, and the deal to think about, it's not 11, because if you have one string and you're cutting up into 2 pieces, you cut it once, right? So it's 1 less in terms of cuts. If I got a string and I'm cutting it up into 3 pieces, I cut it twice. Does that make sense? Okay, so each of these So each blah, 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 like that, represents a round of beta oxidation. And if we look right here, so and it starts from this side out. So we're going to go in this direction. So we start here, and so our first CoA, that acetyl CoA that comes out of it is this. And you stick another CoA on it, and then you take the next one, you stick another CoA on it, you take the next one. Okay. We're going to go through the actual process. Yes?
Going back to the last thing, the name of the cycle, what did you call
Yes.
the cycle?
Carnitine cycle,
Okay.
I guess. I don't know that it has an official name. If it does, it's probably carnitine cycle. All right, so let's go ahead and look at this process. You have the overall process here. So we don't need to worry about zooming out. But you can see you have 1, 2, 3, 4 steps. All right, you're going to be responsible for what the compounds are at each step. I'm not going to make you responsible for the enzyme names. All right, so we're backing off on memorizing stuff. Trying to make it less. And so what we've got here is the first step is it's called beta oxidation because we're going to end up oxidizing the beta carbon. And so it's alpha beta. And what we want to do is we've got this. The first thing we do is we pull 2 hydrogens off and make a double bond. Okay, there, we can see all of it now. So we make a double bond here, which is oxidation, and we take those 2 hydrogens and we load them onto an FADH2. Okay, so we're capturing the energy of those hydrogens, or when we do that oxidation, we're capturing that energy as an FADH2. As we know, an FADH2 is attached to its enzyme, so in this case, this acyl-CoA dehydrogenase, and it can't leave, so then it's going to have to transfer its energy with the hydrogens to a ubiquinol, or ubiquinol to make ubiquinol. And then the ubiquinol can float off to the electron transport chain and be used to make, on average, 1.5 ATP. All right, next thing. We're going to continue to oxidate. We're going to add a water to this, right? We're going to add a water across that bond. And what we get now is we have an OH, right? Okay, so now we have an oxygen for the next step. In the next step, we're going to be able to remove hydrogens again. So we've got these 2 hydrogens, right? We're going to remove them, and we're going to make an NADH, and we're going to oxidize this further by having this carbon double bonded to oxygen. All right, so at this point, we've made a ubiquinol, and we've made an NADH. And then finally, I now have a carbon here, I have a carbon here. I can remove this CoA with the acetyl group to make an acetyl-CoA, and now I have a free fatty acid. And we're going to add a CoA here, and now I have a free fatty acid, attached to a CoA that is 2 carbons shorter. These 2 carbons shorter than the previous one. Okay, and then not the dotted line got cut off or something here. You could connect that dotted line. Then we come up and we take that thing that free fatty acid that's 2 carbons shorter, and we go up and we run it through the cycle again. Okay, until it's all CoA. If it's an even numbered carbon fatty acid and it doesn't have any double bonds on it, this is all we do. We just do this over and over again until we turn all of it into acetyl-CoA. Yeah, Lauren.
One of those is just to make acetyl-CoAs that home through the citric acid.
Yeah, well, it's the point of this is to make the acetyl-CoA, and that one of the things you can do once you've made that acetyl-CoA is you can run that through the citric acid cycle, right? Okay. You could also use the CoA to synthesize amino acids. We haven't talked about that, but okay. And we can also make ketone bodies, and we'll talk about that later, how they're made. But ketone bodies are made, I'll talk about really quick the instance or the reason for it. Ketone bodies are made if you're not getting enough carbohydrates. So if you're not eating carbohydrates and you run out your carbohydrate stores, your brain runs on glucose, okay? And so what your body does, and your heart runs on glucose to some extent, what your body does to compensate for that is it makes ketone bodies from acetyl-CoA. It breaks down your fats, makes ketone bodies, and your liver ships those out in the blood to help fuel the tissues. Yeah.
Is that something to do with the keto diet?
Yeah.
I think so. Yeah, you don't. Okay. Yeah, you're trying to do ketosis. This is ketosis.
It's like diabetic ketoacidosis. Like that's why diabetic ketoacidosis occurs.
So what's going on for diabetes? What's going on is your body you release insulin, but your tissues can't see the insulin essentially, and so they think you're starving and you have no glucose, and so they make a bunch of ketone bodies, even though you've got lots of glucose in your blood anyway. Yeah.
So if the fatty acid already has a double bond, does it just give the first step?
So that's right. If it has a double bond, that's a great observation. So
if it's not in like an even spot, what if the double bond is?
There's a way to move it. Okay, so I'll show you the way to move it later, but I'm not going to make you responsible for it. But if we do have a double bond already, you can start here, right? As long as it's in the right spot. If it's in the wrong spot, you flip it over. But if you do have a double bond, what happens to the amount of energy you get out? More. You get less, because you don't get this bit, right? You're getting a ubiquinol here. If you skip that step, we don't get that ubiquinol that round.
So does that mean like saturated gives more energy?
Yes, and you'd expect that, right? Because this is more oxidized. It has fewer bonds to hydrogen. So let's go ahead and look at. Let's think about let's summarize what we get out of this reaction. So out of one round of. Oh, all right, so one round of beta oxidation, assuming it's a full round, produces one NADH and one ubiquinol. All right, and so if we think about that in terms of average energy from the electron transport chain, this is going to be 2.5 ATP. This is going to be 1.5 ATP equals 4 ATP. Okay? Compared to glycolysis where you were getting 2 per glucose, right? ATP. And then you get what you get afterwards. They're not the same molecule, but this is just one carbon. Anyway, questions?
What was the part about you saying saturated fats give more energy?
Saturated fats are going to give more energy because they are less oxidized. So this whole process, any of these processes in terms of catabolism are about oxidizing. So the fewer bonds you have to oxygen and the more bonds you have to hydrogen, the more reduced you are. So let's think about a super simple fatty acid here. Let's go ahead and I'm going to draw the carbons so we can see the hydrogens. Let's take this one, Okay, that's horrible. But let's draw it like that. So it's a 3-carbon fatty acid, right? Let's go ahead and do the same 3-carbon fatty acid, but let's actually not have the double bond. You see we have a lot more hydrogens on this. So there's more energy to be had from this than that, because this is more reduced than that. And you can see that in beta oxidation because we skip a step once we skip an energy yielding step if the double bond's already there. Other questions?
Like you're skipping step 3.
We are skipping step 1 if we have double bond, because then we make a double bond there and it's already there. Okay, let's see what we can get done in 6 minutes here. So given that, so we know the yield from beta oxidation, calculate the number of ATP that can be made from a, let's make it, I don't know, 10 carbon saturated, actually no, let's make it a 6 carbon, because then it has the exact same number as glucose, right? It's gonna get, the difference is gonna get bigger, but saturated free fatty acid. I'll fix the word saturated. You guys just go ahead and do it. All right, so you should know the numbers. So what we're saying here is that those, the acetyl-CoA that get made go through the citric acid cycle.
Question.
Whoops. So now that you've thought about it a little bit, how many rounds of beta oxidation?
5,
2.
6
We're going to break it into 2 carbon bits, right? There are going to be 3 2-carbon bits and 6 carbons. You've got a string, you want to cut it into 3 pieces, you cut it 2 times. You're welcome to work together on this.
ATP solely from beta oxidation.
No, from the citric acid cycle as well. So let's go with PO ratios 2.5 and 1.5, our usual PO ratios. Let's not make it more complicated. Since we're short on time, I'm going to work it at the same time. All right, I'm going to apologize for my work not being as neat, given we're short on time, not being as neat as I'd like it to be. 11 times 2.5 plus 5 times 1.5 plus, it looks like 3 equals 38? Okay. 38 ATP. Okay, so that's what you should get out of that. If you have questions on it, please ask. Otherwise, you guys have a good day. If you need help,